The following content is provided under a Creative Commons license. Your support will help MIT OpenCourseware continue to offer high-quality educational resources for free. To make a donation or to view additional materials from hundreds of MIT courses, visit MIT OpenCourseware at ocw.mit.edu.
All right, thanks, Marvin. I decided to call my talk Mind versus Brain, Confessions of a Defector. I used to be an AIist. My thesis was reviewed by Marvin. I worked in the media lab. I thought about models of computation that might be more suited to building AIs. Really, I didn't have that many good ideas, although people thought they were good ideas, and this kind of scared me, so I backed away a little bit. And then I found a really cool problem in the area of neuroscience. And I've now left AI at MIT, and I'm a PhD student now in biophysics at Harvard, and I'm working on worms, and I'm trying to figure out how they think to the extent that they do. And so I wanted to sort of go over not really the details of neuroscience or the details of my work. I mean, I'm going to do sort of what Marvin does. I'm going to talk for a little bit, and then we can talk about whatever you want. And if you want to get into the details, that's great. But first, I just wanted to kind of give an overview of, from my perspective, where I see neuroscience and AI sort of fitting in with each other and with the larger context of sort of the history of science and the taxonomy of science. So I sort of self-identify as a mathematician to the extent that people have sort of discipline identities like gender identities or racial or cultural identities. My discipline identity is math. And so I see everything as sort of springing out from that. So on one side, you have sort of the scientific tower where you have physics, and on top of physics, you put chemistry, and on top of chemistry, you put biology, and on top of biology, you have neuroscience. And there's also the sort of computer science where you have, you start from a theory of computation, and then you have sort of software engineering, and then AI. And in both cases, what you're really stretching toward is an understanding of what thought is. And we sort of got to some success in sort of figuring out what the universe is, at least down to a certain level of description. I could turn on the blackboard lights.
Is there one?
Yeah, there is a blackboard light.
I could crank this down.
You don't have transparencies prepared?
No, I don't have. I don't know where to buy transparencies anymore.
I have
That would have been good to know. But
Cut these up.
what's that?
You cut these up.
But what we're really trying to get to is sort of this fundamental question of what is human experience. And human experience is sort of dominated by consciousness or cognition or whatever you want to call it. And we really don't know what's going on there. We have something called cog-sci. And it definitely connects to both neuro and AI. But it's pretty fuzzy right now. And a lot of people take the metaphor of transistors and talking about the brain, and that, oh, if we're, as neuroscientists, we spend a lot of time looking at the details of what happens in the nonlinear regime of this sort of neural transistor, but it really doesn't matter because what matters is when you put the things together and so on, which is a good metaphor. But a metaphor that I also like and I see used less often is that we're sort of, right now, in looking at thought and how its relation to the brain, I think we're sort of where Copernicus was when he was thinking about the planets. In a sense, we had sort of the right basic idea. We have the idea that thought happens in brains, and Copernicus had the idea that planets orbit the sun, which at the time was a new idea for Copernicus, and in relative scheme of things is kind of a new idea for us that thought happens in brains and happens by electrical impulse. But Copernicus didn't have gravity. He didn't have Newton. And so in describing the orbits, he had all of these little corrections and epicycles and deferents in trying to make sense of the things that would later all follow from this very simple theory of calculus and of gravity, but that was yet to be discovered. And so I think that there's something that we're getting to in the realm of cognitive science, some sort of new mathematical insight that I think will be on the same par as the discovery of calculus in terms of how networks emerge to perform complex computation in living systems. And the way that I think about that is when we really get down to the essence of calculus, it's about what happens in the limit of things sort of acting in similar ways as you cut them into smaller and smaller pieces and have more of those pieces, right? And what we're looking at in both in neuroscience and in sociology to a lesser extent, because fewer people are doing quantitative things there, but we're looking at scale-free networks, where as you partition the network at different sizes of partitions, you get the same sort of in degrees and out degrees. And we really don't know what we're talking about when we go into scale-free networks, but it seems that there's something there that relates to sort of the mysteries. The things that we're bumping into that I feel are the same sorts of things that people were bumping into shortly before we figured out calculus. So anyway, that's sort of my big philosophical spiel. I also wanted to tie into this quote from Ernst Rutherford, who's kind of one of the greatest curmudgeons of science history, who once said, all science is either physics or stamp collecting. That either you're writing down the equations and you know exactly how things work, or you're just sort of saying, oh, this looks nice, let me write down what it looks like and where I found it. And biology is definitely still largely in the stamp collecting realm. And it doesn't have to be. It's not the nature of studying living systems, which is the reason that we have biophysics, and it's the reason that I am in a biophysics program, right? But a lot of, there's sort of this cultural tradition in biology that goes back to sort of Darwin, where you just sort of look around the world, you write down what you see, and it's a time-honored tradition, and it certainly gets you pretty far. But it seems to break down when we look at the brain, because you start cataloging these things that are so minute, and you start cataloging them in isolation instead of considering them as dynamical systems that interact with their surroundings. And it seems that you really have a hard time putting those pieces back together once you've sort of collected them in separate observations. So that's another point I wanted to make. And then I was just going to talk a little bit about why I think things are changing, especially in the neuro domain. There's this sort of classic way that you do neuro experiments is you have some sort of furry creature, and you present some sort of stimulus, and then you stick a wire somewhere into the brain, and you measure what happens depending on the stimulus. And you don't know what you're looking at, but you know that you're looking at something, and you can get some sort of idea as to what things are similar. Hubel and Wiesel did these great experiments where they stuck electrodes into cats' visual cortex and basically just kind of waved their arms around and saw that some of the neurons were orientation sensitive, and that's the foundation for much of our current knowledge and research about vision in mammals. But it doesn't tell you how the different things that you're looking at relate to each other. And it gives you only the merest glimpse. I mean, even in the most dense sort of electrode applications, you're going to get a maximum of thousands or maybe 10,000 signals out of this at once. And most likely, none of those neurons that you're looking at are going to be anywhere near each other on the scale of synapses. And so you're essentially just sampling a population. And in fact, there's a lot of research in this field that goes under the rubric of population dynamics, where you're sort of just looking at things in the aggregate. And I heard a nice little metaphor, actually, just today in an unrelated class about populations, where if you're, for instance, examining the population of people walking down Fifth Avenue, in New York City, you can measure things like the average rate that people are walking down, but you're never going to capture something if you're just looking at flow across everything, like every 100 meters people will turn into a shop and stop for a few minutes and then come back out and start again. So you're only getting sort of this very broad strokes, or otherwise, and you're taking the neuron out of its natural context, and you're just saying, okay, here I have a neuron in a dish, whatever, in some sort of growth medium, and I'm just going to probe it and see what happens when I stimulate this neuron in different places. And then you're likely exploring parts of the phase space that this neuron would never experience in the system, and you're getting no indication as to which parts of the phase space it actually is in, or how that would relate to other parts of the system. And when you're dealing with this complex information processing network, that's pretty critical. So that's sort of the classical state of neuroscience, which basically plateaued, I think, about 30 years ago, which is the reason why people like Marvin are pretty frustrated with it. But in more recent times, we've started to get some different ways of looking at neural systems that I think are really exciting. This is why I decided now is a good time to be a neuroscientist. So now we have, if we zoom in to sort of our physicist's spherical neuron, it has a cell bilayer, and neurons are powered by channels which interrupt the bilayer and can pass certain types of ions. And naturally, ions carry charge, and some channels, depending on concentrations and the voltages, will have positive ions flowing in, positive ions flowing out, negative in, out. And these dynamics basically form the basis for neural activity and action potentials and everything. And we found some of these channels, or things that functionally are like channels, in prokaryotes, in basically single-celled organisms, in algae, in archaea, in bacteria even, that have really useful properties. For instance, there's one called channel rhodopsin, which it turns on if and only if there's blue light impinging upon it. And this was discovered about 15 years ago, and about 10 years ago, it was sequenced. And now, with the ability to synthesize genes and introduce them into other organisms, we can take that gene and add it to a neuron, which is not at all where it belongs. Nature would never put this type of channel in a neuron, because there's no reason for neurons to be sensitive to light unless they're photoreceptors in the retina. But now that we have this blue light activated channel, we can point a laser at this cell, and when we turn it on, if it's the right wavelength, the cell gets activated. When you turn it off, the effect disappears. And so you can now do these very precise sorts of perturbations and see what happens. And because it's a genetically expressed channel, what a lot of people did initially is they would just express the channel in a certain class of cells that has a specific promoter that's been discovered, and then just use a wide field blue lamp, light up the entire brain, and then only that class of cells in which you express the light-sensitive channel will turn on, so you can see what does this class of cells actually do. There's a similar channel that's activated by yellow light that removes, well, actually it introduces negative ions, introduces chlorine, and that causes the cell to hyperpolarize or deactivate. So you can selectively inhibit populations based on genetics, and this allows you to do something like a knockout. A lot of people do knockout mice where you remove some class of cells, say here's the behavioral deficit, but you can do it without any, basically you can do it with a positive control, because if you aren't shining yellow light into the skull, then it's just like a regular mouse. And this is really helpful for determining those effects. But even so, this is still population dynamics. You're still just talking about some broad class of cells, all pyramidal neurons, all basal ganglia. Here's what happens when you remove them. So there's another piece of it. There's another piece of this puzzle, which is multi-photon microscopy. And this is something that, how many people know multi-photon or two-photon? Okay, so this is I recently learned about this too, and it's really cool because this is a sort of thing that when you're first like 8 years old and you hear about lasers, this is a sort of thing that you would imagine that you would do with lasers, and then you learn a bit more about lasers and you realize that lasers don't work that way, and then you learn more about lasers. You're like, whoa, actually you can do that with lasers, and what you do is you have one laser. It's a femtosecond laser, which is necessary because of the ridiculous synchronization that's required in doing this. Not only does it have to be a femtosecond laser, but you actually, even though it's 2-photon, you're using 2 beams. You can't have 2 lasers because they won't be well enough synchronized. So you have to split a laser into two beams. And then you focus using fancy optics that I don't know enough optics to draw those 2 beams onto a single point inside your sample. And the wavelength of this laser is twice the wavelength necessary to excite your channelrhodopsin or your halorhodopsin. And what happens is there's a small but non-vanishing probability that 2 photons from the 2 branches of this will arrive at exactly the same point in not quite Planck time, but in sort of molecular excitation time. And if those 2 photons arrive close enough to each other, they'll have exactly the same effect on the state of the molecule as a photon with half the wavelength, or twice the energy, because those 2 photons each deliver that amount of energy, and so it gets doubled. So this only can happen at exactly the spot where the 2 beams converge. And so you get this extremely selective, not only Z slicing, but also an even more selective X and Y slicing. And so you can use this to target an individual neuron, and you can do it repeatably, you can do it reliably, you can do it in any space in your working volume. You can target using basically acousto-optic deflectors, which again, can't draw, so they're going to be black boxes. But you can do it very fast. It's expensive, but you can direct these at hundreds of hertz. And so you can essentially write to anywhere in the brain. You can turn things off or on as you wish, as long as you know the locations. And the other final piece of this sort of optogenetics puzzle is there's a fluorescent protein called GCAMP, which is sort of like a GFP, but with a calmodulin attached to it. And the calmodulin binds calcium ions, and the GFP is a green fluorescent protein. But when the calmodulin isn't bound to a calcium, it sort of hangs out here and disturbs the conformation of the GFP so that it can't fluoresce. But when this binds a calcium, then the GFP fluoresces green. And calcium is one of the major signalings for neuron firing, especially for neurotransmitter release. You need an influx of calcium. So this basically tells you whether the neuron is active. And as if that weren't enough, because it is a second messenger, just this year, there is a protein developed that's a membrane protein, also genetically encoded. All of these can be just engineered into a line of animals, and then you don't need to worry about it anymore. No injections or anything. So there's another one that originally nature intended this as a proton pump. It's archaeorhodopsin. It's a light-sensitive proton pump. But what they were able to do is to silence the proton pumping, basically to disable that aspect of the protein's function. But then they discovered that it actually then has a fluorescence which is proportional to the voltage across the membrane that it would be moving those protons across. So in sum, you can activate neurons, you can inhibit neurons, you can measure the calcium concentration or activation, and you can measure the voltage, and you can do it all anywhere you want very fast. So this is basically a toolkit for doing experiments that you could really only dream of with electrophysiology. In particular, the one that I'm working on right now is the worm, C. elegans, which is a very well-studied organism. And in fact, it's the only organism for which we actually know the complete connectome, which a lot of people talk about connectomes, and it's sort of a dark secret of neuroscience that we already have the connectome for C. elegans and we can't do anything with it. Because it turns out that just knowing where, you have one neuron here, and it synapses to this neuron here, and this controls the body wall muscles. That doesn't really tell you anything like, well, maybe this is an inhibitory synapse, maybe it's excitatory, maybe it's non-functional, maybe it's stronger than the other synapses on that cell, maybe it's weaker. And so it basically gives you very little information to start from if you're trying to understand how this organism thinks or computes. But since we know where all of the neurons are, and there are only 302 of them, it's not crazy to think about using this microscope and these biophysical techniques to actually build a model pairwise, if need be, of all 90,000 pairs of how every neuron affects the behavior of every other neuron. So that's what I'm working on. I think that actually doing these sorts of observations is something that's never really been done before. And again, to make a ridiculously grandiose comparison, it's kind of like how Newton only was able to do what he did because of Galileo developing the tools to observe phenomena that had never been seen before. And I think that advances, well, this is actually a quote from Sidney Brenner, who is the first person to suggest that you can study this worm and maybe learn something from its nervous system. Advances in science usually come from new techniques, new discoveries, and new ideas in that order. And so now we have the techniques, we're working on the discoveries, and the hope is that it will lead to new ideas. So, questions? Yes?
So you've given us this overview of like the state of the art right now and possibly how it's going to be in 5 years, but how do you think neuroscience is going to be in about 20 years or maybe 30 years from now?
Well, what we can do right now is this most basic organism that nature has to offer. And the natural thing to do once that gets solved, which we don't know how long it'll take because we don't know how much detail is really important, but I think that we can solve the worm in 3 or 4 years. And then the next step is the zebrafish, which is also optically transparent, which is handy for using these sorts of optical microscopes. But the zebrafish has 100,000 neurons, so it's a big jump in complexity, and it has a lot of the same sorts of brain regions that you see in mammals and even humans, although they often go by different names. But it's a similar structure, it's a vertebrate, and it has eyes, which the worm doesn't. So that'll be the next thing to look at. And I think that'll probably take another 5 years or so. And then maybe Drosophila, bees are pretty complicated. That's the first place where you get something that resembles language. So that might be really interesting. And eventually, mice, cats, dogs, monkeys, and humans. And definitely the path that this goes on in an ideal world is toward taking an individual human brain and turning it into a model. Yes?
What would you consider to be solving the worm? How much do we need to know about this? What are the big problems there? What are the things that we still have to understand?
The way that I have set up the criteria, there's a big list of publications, basically, of behavioral results. And there's a lot of stereotype behaviors. There's something like 30 or 40 different conditions where you can put the worm in these conditions and they exhibit particular, omega turns or reversals or things like that. So that's sort of the baseline to say, well, if you put the worm in the virtual worm sort of in these conditions in a virtual simulated petri dish, they exhibit all the same behaviors. That's sort of your first order check. And then the next step is, well, what happens if you remove a neuron? You pick one of the 302 neurons in a physical worm, you can ablate it with a laser, you can kill a cell specifically, or you can just inhibit it with a halorhodopsin. Then you can see, does the virtual worm exhibit the same behavioral differences as the physical worm under that condition? And you can also do larger scale sorts of things. You can say, well, what happens if I were to activate this neuron 10 times a second every 5 seconds, and how would that change things? So you could do lots of different perturbations, and that to me is the best way to check that you have what I consider a biologically relevant model. But what you're looking for is not anything on sort of you're looking for observables, basically. And I think that's what you have to do, kind of, if you're doing science. You have to be looking at what's observable. And right now, what's going on inside the synapses is not observable. So I'm not going to be simulating that. And part of the hypothesis, since I'm doing this as a PhD thesis, it has to answer a scientific question. And the question is, can you capture the qualitative aspects of behavior as viewed externally without modeling what's going on at the molecular dynamic level? Yeah?
A question on connectomes. So in my understanding of the human brain, I thought that the neurons could grow new connections with other neurons. So in that sense, it's like the map of the connections between all pairs of neurons is constantly changing.
Yes.
So how would that work when we're trying to find the connectome of like a more complicated organism whose neurons do make new connections?
So C. elegans, nicely enough, doesn't do that. But you're absolutely right. Mammals do. Mammals do form new connections. And there's also, even in C. elegans, there's a question of development. It's been shown, not conclusively, but fairly convincingly, that electrical activity is not only important for cognition, it's also important for development. And if you introduce genes that basically only break action potential function of a neuron, those neurons, as they develop from birth onward, they don't form the connections that they should. And so there's something going on, there's some computation going on there that's development specific, probably, because in most areas of the brain, once you reach a certain level of maturity, those sorts of processes stop growing. So there's some sort of computation there, and I'm explicitly leaving that out, because I want to graduate in a reasonable amount of time, saying, development, future work. And it's future work. And that's at the same time as, hopefully, this is a success. Someone will go look at the zebrafish, and someone will go look and try and figure out how C. elegans develops from a larval stage to an adult with all 302 neurons, and how they find each other to connect. And that's definitely important, because how the nervous system develops gives us some clue as to what the functional organization is, because the things that develop in concert and sort of stem from the same developmental program, in a sense, probably have the same functions when they're finished developing. But separate from development, there's also this question of learning. And if you do just capture a connectome, it seems to me that there is a possibility that what you could wind up doing is sort of capturing a connectome frozen in time. You could wind up with some anterograde amnesia effect, because if you're missing some aspect of plasticity, on a short-term scale, you would get the same sorts of responses, but you wouldn't get the same sorts of changes over time. So that is a possibility. The way that you can get around that is if you have tools. Again, we're talking 20, 30 years in the future. These are pretty recent. Who knows what we'll have then? If we can visualize something either at a lower level in terms of what's going on with transcription factors, or if we can visualize how the axonal processes grow, then we can build models of that in the same way that now we can build models of sort of the steady state dynamics in the sense of short time scale dynamics of electrical activity. Yeah?
So are you saying, so how many neurons can you look at once with that focusing?
So it depends on how many lasers you have, right? Number of lasers over 2 equals the number of simultaneous observations, but you can direct the lasers at hundreds of hertz. And so if you want to look at 100 neurons at 30 hertz, you can do that. You just have to multiplex them. And because the things, especially in C. elegans, are on a fairly slow time scale, because C. elegans doesn't have action potentials, or at least they're not thought to be significant for computation, it's doable. As technology gets better again, you'll be able to scan faster. There's actually people working on just fancier optics tricks that let you scan faster, and also you can just use more lasers. I mean, there's nothing to say that you have to be pointing one laser. You can multiplex much faster if you're talking about pulsing the lasers on and off, because they're femtosecond lasers. So the more lasers you have, the better. But ultimately, when we're talking about systems like mouse, where you have to penetrate through millimeters of tissue to get to certain regions, it's probably not going to be optical. I mean, at least it's not going to be visible light. One potential direction is called magnetoencephalography. And when there's a neural current, it induces a magnetic field just by Maxwell's equations. And right now, if you have sort of superconducting magnets, squids, you can detect the currents of order of 1,000 neurons activating at once. So you have to have like that level. But when you're talking about 100 billion neurons in the human brain, that's still pretty impressive. It's pretty impressively fine-grained. It's much better than an MRI. And as time goes on, again, hopefully those things will continue to evolve and improve to the point where you can measure what's going on at a very low level. And then on the control side, similarly, we have transcranial magnetic stimulation. And that also is rapidly increasing in resolution and accuracy. So good.
Do you ever name the worms?
A friend of mine suggested Ellie for C. elegans, but I haven't come up with any others. Yeah?
Okay, so this question is currently not super well formed, but I'm going to
No problem.
Okay, so clearly since you're devoting a few years of your life to this and doing your PhD on it, you do think it's important to try to understand the low-level physics of it all to understand the mind and thoughts, right? Is that what's going on?
So I think what I'm trying to establish is a lower bound on what's important. Because there's a lot of people out there, like Terry Sejanowski, who argue that what's going on inside the synapse is important. The mechanics of vesicle diffusion is important. And if you're not keeping track of the vesicles, you're really missing the point. And so what I'm trying to do is, at least for one organism, for 30 behaviors of this organism, say, you know what, vesicle motion is not important for this. And then hopefully, once that's established, we could sort of move forward and say, okay, well, maybe the neuron, the different compartments in the neuron aren't important either. Maybe there's some functional units that we can start to consider. But I'm trying to just sort of establish that lower bound. And in addition, I think in C. elegans, where you have a total of 302 neurons, that's on the same order of magnitude as the number of sort of functional regions that we've identified with MRI in human brains. And I think in C. elegans, each of the neurons really is pretty specialized to do a certain job in the organism. So I'm not sure that you could go that much higher in this model system. But at least I'd like to say the neurons are the lowest level that you need to worry about.
Okay, so as a follow-up to that, so that's really cool. That actually clarifies some things for me. But do you think that work on higher level stuff is still useful at this point?
Oh, yeah.
Okay.
Yeah, absolutely. But I think that work on higher level stuff is largely the same sort of work that has been possible for a while. I mean, there's certainly an argument that we have way cheaper and more powerful computers than we did when people started doing AI. But I feel like most AI is not really about scale. Unless you're talking about Google-style AI, which I feel like is not really the point. There's the reason that I moved into neuroscience is because it's clear to me that there's something that you can do now that you couldn't do before. There's something you can see that you couldn't see before, and so there's got to be something that you can learn from that. I don't think that this is the most probable path to intelligence. In the sense, I think there's many paths to intelligence, and the collection of all of them that don't involve looking at brains at all is a greater probability of success than the collection of all of them that involve looking at brains. But I think this specific path is the most probable single path. If you were to compare it to hierarchical temporal memory as a specific way that you can go, or if you were to compare it to Bayesian networks as a specific way to go. I think that this is more likely than any specific thing. So it's not that I think that it's the best, but it's certainly the clearest at how to proceed, and I like that. Marvin.
Is there an estimate of how many genes control the nervous system in the worm?
In the worm, I don't know that number. I think if you're talking about just like channels and transporters, it's probably something like 100, if that. There's a class of genes called UNC for uncoordinated, and when you remove those genes, the worm doesn't really swim very well. And there's about 120 genes in that class. So I think that's sort of roughly the nervous system genes, if you will.
Yeah, I've seen estimates for the mammalian brain, which are 20,000.
Well, 20,000 is how many genes there are in a human, but maybe all of them are important for the brain. Who knows? Yeah.
When you're talking about removing some of those neurons, are you talking about doing that in a mature worm that doesn't have more development? Okay, so
Yeah.
What about if you do that in a worm that hasn't developed yet entirely? Would you see things develop, new connections?
Things get screwed up. If you do it in a mammal, things adapt, and you wind up kind of being okay. The worm developmental system is not that complicated, and if you start killing things in the larval stage, it kind of just isn't happy. It'll usually live, but the neurons that were supposed to go there will just sort of get lost.
Is there a tipping point for what animal is complex enough to adapt versus what isn't, like the worm?
So the word for it is non-eutelic. Eutelic means that the network structure is fixed and it won't adapt. And I think you can get up to the level of about a snail, and there are some eutelic snails. And then beyond that point, certainly all insects have adaptive neural networks. Yeah.
Could you just describe a tiny bit more detail the kinds of experiments that you're doing in the short term? I mean, you've given some general sort of things about the method, about the experimental techniques, but what are the sorts of experiments you're doing in the next time?
So the nearest term thing is actually just to focus on the readout, which is the calcium image readout, to express in all the neurons, which no one has ever done because there's this, again, it's sort of a cultural bias. Not that I'm unbiased, I have one bias, and biologists have the other bias, which is to isolate the smallest publishable unit, sort of say, okay, I'm going to work on this cell and figure out its function. Anything else is noise. And so you try to minimize the expression of your transgene. And what I'm trying to do is maximize it. I want it in all of the cells because I want to be able to capture the entire system so that I can treat it as sort of a closed system with well-known inputs and outputs. So in this case, the first thing I'm trying to do is just to express calcium, just to do confocal imaging, and to see if there's any patterns that pop out. And right now, I'm just sort of in the process of trying to get this gene to actually express in all of the neurons. Yeah?
So if every worm has the same number of neurons and they're all connected in the same way, what accounts for the functional differences between worms? Say, like, what makes one more uncoordinated than another?
Oh, so when you do have those mutations, you do get different network structure. Well, not all of the time. Sometimes it's not different network structure. Sometimes it's just that a certain class of neurons is not excitable. It won't fire because it's missing voltage gated channels or something like that. Actually, none of them have voltage gated channels. But if it's missing receptors, for instance, it'll just sit there and it'll be a roadblock to signals that are supposed to go through there. So yeah, when you start mutating, that breaks the rule that they're all exactly the same. Yeah?
Follow up on that question, how certain are we that adding genetically these kind of gates changes the topology and our genetically modified worm will behave in the same way, in the same manner as an ordinary worm?
It's a very good question, and the way that I've sort of dodged that is to say what I'm looking for is sort of repertoire of 30 or 40 behaviors. And so suppose that introducing all of these foreign channels really does change what's going on at the level of neural dynamics. But suppose that when you look at what the worm does under various experimental conditions, it's still the same. Then, yes, you're going to be simulating something that isn't natural. You're going to be simulating the modified state with whatever dynamical changes are introduced. But you're still going to be capturing the computations that lead to the same behavior. And so in some sense, if you can't tell the difference right away, and what you're trying to do is not to be able to tell the difference to your model, then it doesn't matter if those changes are introduced. But there is definitely a risk that some of the behaviors drop away. For instance, with the voltage sensor, as I said, it's originally a proton pump. And if you put a proton pump into a neuron, you're not going to get any spontaneous activity because it'll just depolarize, it'll hyperpolarize all the time. Well, all the time that channel's being activated. And since it's supposed to be a passive sensor, that's not good. So it's critical that this sort of performs as advertised by the people who engineered it, that it doesn't perturb the neuron. But if it does, you're going to know. And it's not going to do the things that it's supposed to, the worm won't do the things it's supposed to do. Yeah?
So do you think that worm's behavior will just be a collection of if-do sort of rules, or will it be a bit more unpredictable than that?
So I think my intuition from what I've observed so far is that it's a collection of largely autonomous local control loops with some long-distance modulatory connections that are usually not active except in exceptional conditions. So you're going to have in each body segment just a tiny control loop. In fact, there's some evidence that the control loop consists of one cell that just happens to synapse onto muscle and also be a sensory neuron that basically says, if the body segment ahead of me is stretching this way, then about 50 milliseconds later, then I should stretch that way too. And so this sort of propagates the undulating wave. There isn't a pattern generator, for instance, like you would see in a higher organism that sort of says, okay, you. They sort of coordinate with each other. And then when something goes wrong, then there's other connections that seem to get brought into the loop and modulate those typically autonomous systems. But I don't know. I expect that many surprises await in the full model. Yeah?
What do you think about the current efforts to jump directly to the connectome of, say, a mouse? Do you think it's possible that these projects will succeed without insight?
Without?
Insight.
I think that it's possible that they will succeed and not provide any insight, if that's what you mean. It's certainly physically possible if you had enough time and resources to do electron micrographs of an entire mouse brain. I mean, it's not that far off. If you had, say, $80 million, you could just do it, right? It just takes a lot of microscopes and a lot of time. So it's not that it's not actually a very high risk project in that sense, but it's also not really that much of a high reward project because no one knows what to do with that data. Like the most advanced, I actually did lab rotation with Jeff Lichtman, who did the Connectome project, and what they're looking at right now is they're looking at, well, when axons synapse onto dendrites, are they choosing which dendrite to synapse onto randomly or not? And that's a sort of analysis that you can do on that data. And it seems pretty obvious that they're not random. Right? There's some sort of pattern to how things connect in the brain. Like, I think that's intuitively clear, but there is still also this community of people out there who say, no, you know what? We've done neural networks with random connections. They seem to be pretty clever. They're just as clever as the neural networks we've built with carefully designed connections. So, there's no real reason that the brain needs to have specific patterns of connection, and nature is always parsimonious and efficient, so it's probably random. And so this is the sorts of stuff that you see in connectomics, and it's not exactly what I'm interested in.
What sorts of inputs and outputs does the worm have? You mentioned muscles and...
Yes, so the outputs that we can observe are pretty much all muscles. There are body wall muscles, there are egg-laying muscles, there are anal muscles, there are head muscles. And then the inputs, there's a few light-sensitive neurons. So it can do sort of it's actually very much like a QAPD, which is the sensor that was put on top of early heat-seeking missiles. So it has just enough information that it can navigate toward light or away from light. It has a large variety of chemical sensors, or basically odor sensors. So that's how it finds food. It also has chemosensors that aren't classified as odor sensors, like carbon dioxide sensors. So if there's too much carbon dioxide, it goes away from there. It has touch sensors, so if you poke it, it goes away. I'm trying to think some of the other sensors. It's mostly odor, definitely by cell count. Odor, touch, and light. I think that's about it.
So in the simulation, do you intend to just stimulate the inputs and try to get some expected outputs, or are you going to brute force all possible neural states for the worm?
So it's 302 neurons, right? So if each neuron has, say, 2 states, it's a lot. It's a big number. I'm not going to do that. There's a hierarchy of different sorts of levels of data collecting pain, and that's the top of it. Well, actually, the top of it is that the state of each neuron is a real number. And you have to sample a vector field, to the precision of its curvature, 302-dimensional vector field, and then it goes down from there to at the very bottom. You just start saying, okay, each of the neurons is a linear function of the other neurons, and we just have to put together a 302 by 302 matrix of synaptic weights. And in fact, it's known that there's only about 7,000 synapses, so you can zero most of that out right away. And there's only 7,000 numbers you need. That's sort of the hyper-optimistic point of view, which is kind of what I thought going into this. It's probably more complicated than that, but I think there's at least some separability where you can say the synapses and global peptide diffusion are basically the only ways that you can have an impact of one neuron on another. And you can do some sorts of things from genetics just to say, well, here are the channels that are in there. You aren't going to have anything that can't be explained in terms of a potassium current and a sodium current and a calcium current. And so there's definitely prior information that you can put into it from the connectome and from genetics. And you can even go so far, and people have, as to put in prior information from what you would like to get out of it. You say, well, I want to evolve the synaptic weights towards something that exhibits this type of undulation. And lo and behold, it exhibits that type of undulation, but then you don't know whether it correlates to reality or not. But if you have an instrument where you can check, does this correlate to reality, then you can still use those sorts of approaches as shortcuts through the sort of state space of possible neural networks. Yes?
What's the life cycle of a worm like? How long does it take to create a worm that has been genetically?
It's wonderful. The life cycle is 4 days. Yeah, it's very convenient. And it's a self-fertilizing hermaphrodite too, so it clones, by itself. It's pretty good. It's also the only organism that cryogenics has proven to work on. You can put worms into liquid nitrogen. 20 years later, you can thaw them out and they start crawling around. Yeah?
So you said that we hope that the neurons have basically a binary state. What's to say that they don't have 10 binary states or something?
Yeah, no, I don't think they do have a binary state, and I think it's sort of, again, in some ways, it's not the right way to think about the problem that it's the sort of that the neuron has a state, because it seems like a lot of what's going on is, especially in C. elegans, because we're essentially talking about analog computation because there aren't known to be spontaneous action potentials. What you're really looking at is more like a control system. And so some input, like for instance, there's a paper just published this month that showed that certain networks in C. elegans are there to compute time derivatives. And I believe that that's just a functional block of a PID controller. Right? Then we're going to find the integral part, and then something that linearly sums them and then feeds back. So I think it's not really so much about finding the set of states and then saying, okay, for this state you go to there, in the sense of sort of an if-do rule, because I think that it's a lot simpler than that. In that there are just simple equations that govern most of the processes and how they relate to each other and to the environment. That's my hope. The adults can grow to be about 700 or 800 microns long, and about the diameter of a hair, about 50 to 100 microns in diameter, depending on stage. And they're transparent, so they're challenging to spot with the naked eye, but you can, in the right lighting conditions. Yeah?
I remember in the first days, Brenner actually bred small ones.
Mm-hmm.
And for example, I think the nervous system and the gut are separate pretty much.
Right.
And with selective breeding, he found some where they were in the same plane,
huh.
and half the length and so forth. And the purpose of all that was so that he could get a whole worm into the target of his electron microscope.
Uh-huh.
So he, that way he had single pictures of the whole thing.
I see.
And, it sounds like they don't have to do that anymore, but it was an interesting case where they controlled the evolution of this beast
to be easier to study.
And that's not a bad idea.
No, it's not.
I don't know if they reduced the number of neurons by accident.
Yeah?
What percentage of this effort do you think is going to be devoted to combining all of these tools, and what percentage do you think is going to be running the aggregate to learn something?
I think once everything works, the process of taking a worm and sort of scanning it in will only take a couple of hours. It's so 99.99% building the tools, because really that's about how long you have before these sorts of the dyes get photo bleached and the laser power starts to heat things up uncomfortably for the worm. And then you're studying a different system. Yeah.
Who funds this research?
Larry Page.
Personally, or?
Yes. Yeah, the NIH isn't a big fan. Although there was a faculty member who put in a valiant application. He managed to tie this research in plausible ways to a cure for Parkinson's, which I was really impressed by. But the NIH was not so impressed.
Did you guys apply to a grant from Larry? Did someone talk to him?
It was his idea, actually.
Oh.
Well, I mean, he wasn't the first person to have the idea, but and in fact, he wasn't even the first person to tell me about it. He was the third person to tell me about it. But when he told me about it, I listened. And then when I decided that I was going to do it, I said, well, not to him, obviously. I went through, my network of many people, and eventually got a message through that, I wanted to do this for real, and, could he spare 0.0001% of his fortune to make it happen? And yes.
So how far along are you? How many worms do you have?
So it's not anyone's number one priority to make these genes, again, because all the people who have the skills to make the genes and breed the worms to express them are naturally biologists who have this bias towards, no, we want to do small systems. And so I have to figure out how to do it myself, and not having any background in biology, most of my time lately has been spent taking classes in biology. But I've also been working on some of the computational tools that we'll need. For instance, when you have a worm in this sort of wobbly conformation, and it's changing, obviously, as it behaves, you need to find the neurons and track them at rates of at least 100 hertz in order to keep your lasers pointed in the right place. And so I've been working on that, and I have some pretty good algorithms for that, as well as isolating the signals. And basically, when you're imaging something in confocal mode, which is going to be the first thing that we do, is a lot cheaper than two-photon, is you have to separate neurons that are on top of each other. They have different signals, and the way that I'm doing it is to say each pixel is some roughly linear function of some set of the neurons that are near that pixel in 3 space, and you can actually just use a singular value decomposition followed by some simple heuristics derived from Microsoft Paint to say, here's where you just literally you flood fill in where the neurons are. And it works incredibly well. Yeah?
So if you had like a multiple state process or something, like not just that you wanted to look at in the worm, like something, like if you wanted to look at all this neuron as it's producing like a new clone or something, like that multiple step, would you be able to?
Sorry, what do you mean by multiple state?
Yeah, like instead of like a stimulus and response sort of thing, if you wanted to measure something that, like a process that took longer.
Oh, yeah. So the nice thing about having a control over all of the neurons is that you can also control the sensory neurons and thereby put the worm into the matrix. And you can make it experience whatever you want, in principle. This has been done with zebrafish. So the way that you do this is you use a myotoxin to prevent any of the muscles from contracting. And so then you have a perfectly still, paralyzed animal. And then you can feed it whatever sensory stimuli you want. You can read out the motor stimuli and motor responses. You can feed that back into your simulation. It's exactly like the Matrix, and then the animal is convinced that it's in this environment that doesn't exist. Yeah.
One thing that I don't, perhaps I'm just being a little bit not picking up on all the details here, but one thing I don't understand is how much of the state of the worm, so it's 300 odd neurons, how much of the state are you, do you have knowledge of at any particular time? Is it all of those neurons?
So again, it is, you can measure, the way that it works, if you have one pair of lasers, you can measure any neuron at any time, and the measurement process takes about a few milliseconds. And the time constant of these neurons is on the order of 40 or 50 milliseconds. So you have to be a little bit smart about which neurons you want to look at when. So in that sense, you can't look at all of them simultaneously unless you have more lasers. Again, more lasers solve everything. But yeah, so you can look at calcium, you can look at voltage, and you can't do those at the same time either. You have to say, okay, I'm going to read the voltage of this, I'm going to read the calcium of that. And there's actually a theory called optimal exploration of dynamic environments, which was also just published this year, about a way to algorithmically make that decision of what thing do I want to look at that is most likely to lead in the long term to gaining the most information about the system, given my expectations of what it might look like.
And then conversely, how much of the state can you affect?
And it's the same, you can perturb, either stimulate or inhibit any neuron at any time to any intensity. So you have a little bit more control there. But again, you do have to multiplex, and you have to take advantage of the fact that the neurons are not going to react as quickly as you can.
And then for each cell, how much of the state do you think you accounted for?
You're basically just looking at two numbers, but you're looking at those. You can look at those numbers anywhere in the cell. So you don't have to assume that the cell is isopotential, although it probably is for most of the neurons, not for the ones that run the whole length of the worm, but there aren't as many of those. So you can collect, in some sense, as many numbers as you want if you're interested in looking at all of the, gridding a particular neuron to a lot of different locations, and you don't care about looking at anything else. But you are only going to be looking at calcium or voltage, at least with the proposal that I've got here. There are other genetically encoded sensors, but as far as I know, none of them are likely to be nearly as relevant to neural activity. At the same time, again, you're not getting what's going on in the synapses, you're not getting phosphorylation, you're not getting methylation, and all of those things are certainly important for learning and plasticity, but since C. elegans doesn't have that much of it, it might be okay.
So you haven't got a complete state transition diagram of the dynamics of that?
Right. Yeah, like I said, a complete transition diagram would take years to construct, and the life cycle of 4 days, it's not going to work. Yeah.
Just wondered about your thoughts about higher level questions. For example, if you look at a neurology book, it will explain on the basis of what happens when people get a concussion, that short-term memories are stored in hippocampus or amygdala, I forget sometimes, and they can last there for 20 minutes or so. And then over the next day, a memory trace is copied into some other part of the brain, like parietal lobe or frontal lobe or something. And I've never seen even a paragraphs about, well, how does it figure out where to put a memory and how are memories represented? So a nice question would be, if you take something like the idea of K-lines, the kind of methods you're describing might be nice for that, because the usefulness of a K-line might be that it's a bunch of neurons which go several centimeters. And so if you could look at neurons in 100 places, a centimeter apart, which is very low resolution, then you might be able to find evidence for correlated activities. related to some stimulus or whatever. So some of these techniques might work on a much larger brain just because the increase in size is liberating. It means that the instrumentation can be clumsier if it's looking at whole bundles of fibers.
Right. Yeah, that's what Ed Boyden is starting to look at now. Basically, I think he's calling it optodes. I'm not sure if he coined that phrase, but the optical equivalent of sticking an electrode into a brain is.
What is he using, humans or?
Well, he's doing mice, and he's working with people who do monkeys. The problem with humans is.
Mice have a very nice paper thin skull.
Yes. Yeah, humans have a thick skull. That's part of the issue, but the bigger issue is that the only reason that we get to stick electrodes in humans at all is because it's an approved treatment for epilepsy. Now he's working on getting optodes approved as a treatment for, well, first for blindness, which is kind of an obvious thing. If you can use an adenovirus to express opsins in an eye that doesn't have them, it probably doesn't affect that many people, as most blindness is caused by other things, but for those that do, it's a very, very obvious intervention. Then also PTSD and other sorts of diseases. So as soon as it gets approved to treat any sort of disease, then you can piggyback on that to do human research, but that hasn't happened yet.
In the early 60s, there were some successful experiments. There was a guy named Brindley who not sure what his profession was, but he made some of the first electrodes, and he actually got permission from his secretary, who was blind, to put a little plate with 64 electrodes on her occipital hole.
Wow.
And put little currents in, and she could recognize visual patterns. Each of these electrodes he described as being a little bar, which was about half a toothpick at arm's length. And of the 64 electrodes, about 30 of them actually produced these, and the others didn't work. And so he did that, and then he removed them because it was a pretty risky thing to do anyway.
Right.
And at the time, we had a great neuroscientist here named Warren McCulloch.
Mm-hmm, mm-hmm.
And he got Brindley to come over and talk to us. Incidentally, Brindley later discovered the use of nitrous oxide for producing erections in human males. And he gave a demonstration of that in a famous lecture. So, at the time, McCulloch said, if you're interested in stimulating vision in the human brain, you better do it in the next 5 years or it will be illegal.
Mm-hmm.
That was in the early 1960s. And this was the same time that the worm that Brenner was, and we actually thought about that and decided not to. But anyway, it would be nice if we could get back to that.
Yeah.
And it might be that low resolution things distributed very widely would also give a lot of new information.
Yeah, I think there's a lot of promise in MEG, especially because when you're just sort of putting things on the surface of the head, there aren't issues. It's not surgery, even if you're performing the same effective perturbation to the neurons. And what people are doing with transcranial magnetic stimulation is it's not that great, but it's certainly promising.
And there might be some way of it getting things into cells that actually synthesize proteins,
right,
encoding data and
right,
put them out. bloodstream later.
Yeah, although there is a group working on that. They're calling it the molecular ticker tape, where you basically, it's complicated. I don't think I can explain it properly, but there are a lot of people who are looking for problems to match the solution of high throughput sequencing, where you can take huge amounts of DNA and sequence it cheaply now, and that's one of those. But it would take a pretty heroic effort to then correlate those with the actual experiment that you performed after you've extracted them.
They'd have to say where they came from and how long they took.
Right, and I asked them, how do you identify the barcode the cell? I'm like, well, we don't know. We'll figure that out eventually.
Danny Hillis and I once consulted for Schlumberger, who made instrumentations for oil wells.
Mm-hmm.
And when they have a deep oil well, there's a pipe that's a couple of miles long, believe it or not.
Mm-hmm.
And they get one bit out every 5 or 10 seconds
Uh-huh.
by putting pressure pulses in.
Right.
And we designed hideously elaborate things that would punch tape, and they would come floating up a few days later. It was fun going to the meetings where the geologists explained why each of them wouldn't work.
Mm-hmm. Any other questions? They don't have to be about neuroscience.
Thank you very much. Yeah, go ahead.
Oh, I was just curious, you focused on the experimental side.
Yeah.
Reading out from the worm. At some point you want all of that data to drive a computational model.
Right.
Can you build a computational model now and initialize it in various ways and look for any kind of behavior at all? Or do you absolutely need this biological input to begin to drive the computational system?
I think I'm working on some leads for building computational models. There's the obvious things to do have already sort of been done in that department. Like I said, using genetic algorithms to find parameters that satisfy certain conditions, and it doesn't seem that enlightening. So I haven't pursued it too much, but I'm looking at, and this is actually another really interesting to me connection in terms of AI and neuroscience, is that when I'm looking at building the computational model that tries to interpret this data, the key idea that keeps coming back is critics and selectors, because you need to have some set of possibilities and you need to have some heuristics for determining, based on what data is streaming in, what sort of model seems to apply to it. And then you need to have a meta model where you need to build layers of reflection, where you're saying, well, we need to modify this in this way. And it's not easy to implement that from scratch. So I'm thinking about it, but I think that once we have real data, it'll be more clear what needs to be done to simulate the processes underlying it. And again, it's just sort of my hope that in observing something that has not been observed before, that some insight will come out of that process.
Is there a behavioral diagram somewhere? For example, if you feed it a lot of food, presumably it will stop eating.
Right. Yeah, no, I don't remember the name of the fellow you mentioned who spent years studying seagulls, but Tinbergen, right. There isn't a Tinbergen of worms, unfortunately. All of the studies are, again, it's this very stamp collecting approach, where you're saying, okay, at 24.6 degrees Celsius with OP20 growth media and with worms that are 3 days into their life, and this number of worms per this area of plate with this brand of agar, here are the behaviors that we see in response to this stimulus with this number of milliseconds between repetitions and so on. And no one goes so far as to make a plot with more than one variable, because goodness, what's your control? And it is a little bit frustrating that we're going to have to build those sorts of things ourselves, but hopefully that will be the less hard part than building the model. Yeah?
Could you just make some thoughts about the way mathematics might relate to, I mean, you said you were interested in that subject, so just...
Sure. Right, relate to?
Well, to the theories of simple animals and things.
I think, as I said, I think there's something missing in mathematics. There's some sort of theory that follows from some sort of symmetry that shows up in nervous systems and not in many other places. It maybe shows up in societies as well, but I'm not certain of that. But as far as math that we know, certainly nonlinear dynamical systems is the obvious one, because a neuron is a nonlinear dynamical system. And I think that in simple animals, especially in C. elegans, most of the computations that we see are also nonlinear dynamical systems. As I said, they're integrators or they're derivators, things of that nature. And I think a lot of them will turn out to be amenable to an analysis as sort of differential equation systems. But at the same time, I think figuring out certainly more complex organisms will require a new way of thinking about how to put together a computation. Von Neumann in 1956 started working on a monograph to accompany a series of lectures called The Computer and the Brain, in which he would discuss the differences and similarities and how he thinks that ideas from computer science will relate to neuroscience. And this was 1956. Unfortunately, he got bone cancer, and he died in 1957, and it left the document unfinished and never gave the lectures. And the document concludes very dramatically with this sentence, however, if the brain uses any sort of mathematics, the language of that mathematics must certainly be different from that which we explicitly and consciously refer to by that name today. And that's where it ends. Yeah?
Do you have an idol?
What?
Do you have an idol? Like fictional or non-fictional?
Von Neumann would be the closest, yeah. I mean, Iron Man, but that's just obvious.
So you're over Edison?
What?
You're over Edison.
Over Edison. No, Edison's pretty cool too.
Neumann was my hero because when I finished my thesis, the math department was on neural networks, and the math department didn't know what to make of it, so they gave it to von Neumann.
Mm-hmm.
Did I tell you this story?
No.
And said, "Is this mathematics?" And he said, "If it isn't now, it soon will be." And I got my PhD.
Can you talk about any of the projects before yours that people have already used these tools?
Yeah, so let's see. In terms of optogenetics, I don't know, there's the one, I think probably the most famous one, just because it has a really cool movie, is someone found a promoter for a class of neurons in mice that is coupled to right turns. And so you can put the mouse in some environment, it behaves. You turn the light and it starts turning to the right, no matter what it's doing. And it just turns around and starts going in circles. And you turn off the light and it goes back to what it was doing. It's mostly cool stuff like that. But it's also found use as a replacement for electrophysiology. Anything that you could do by using micropipettes or microelectrodes. Well, to some extent, depending on what kind of time resolution you need or what kind of manipulations you want to perform. But a lot of the things that you previously would need very precise and expensive equipment and calibration for, you can now do much more simply by using genetics and a blue LED. So there's a lot of things like, for instance, even in C. elegans, a lot of work has been done by Corey Bargman in recent years using calcium imaging to just sort of explore more quickly and more thoroughly the dynamics of sensory neurons, which is important to me because if I'm going to simulate the sensory neurons in some pattern that reflects a virtual reality, then I need to know how that pattern would relate to the real reality that they're observing. And so Corey Bargman has done a lot of experiments where she just sort of flows in an odorant, for instance, and she's got a laser pointed at the neuron that's known to sense that odorant, and just characterizing the dynamics of how the neuron responds and habituates, which is something that you could do with a micropipette, but the worm is so small that you need, it's just really hard to get something in there onto the specific neuron that you want and have it stick. And it's been done, but it's only been done a few times by people who are very skilled and very lucky. And these types of optical tools make it a lot easier to do those sorts of experiments, if nothing else.
So you can't, this is a demo, I think, so you can't like change anything about the worm once it's already in there. Like once it's already under the lasers, you can't like add more stuff. But why would you want to?
Well, I mean, there are reasons that you might want to. A lot of experiments are done. It's actually really creative what a lot of cellular neuroscientists do, because they realize that once you have something stuck into the cell body of a neuron, you can introduce whatever kinds of molecules you want. And there are certain molecules that are selective blockers of certain channels. And so you can do things like, okay, what if you don't have any potassium channels? What now? And you can do those sorts of perturbations when you have a physical connection to the cytosol, which you can't do optically. So those, again, are things that I hope to show, but it is not known, that you don't need to do them in order to model behavior at the scale of the organism. Yeah?
Does the worm have any chemical triggers that are controlled by basic organs? Like in humans, there are different glands.
Yeah, so there aren't really glands in C. elegans, but there isn't a circulatory system either, but there's sort of like a shared body of fluid through which waste is channeled that it contacts a lot of cells. And there's some evidence that there are a few neurons that do diffuse transmitter into that, thus affecting a sort of global change in excitability. So yes. The way that manifests in a model is basically just as an extra node, saying this represents the sort of global concentration of glutamate or whatever.
Thank you.
Thank you.
What's the simplest animal that does a little bit of learning?
C. elegans does a little bit. I mean, it's really just associative learning, so it can learn aversions or attractions to temperature or to chemical stimuli or light. But it's probably just one synapse that represents an aversion to this thing, and that synapse changes in strength or something like that. As far as I know, and I'm not a zoologist, so I really don't know. But as far as sort of the set of classic neuroscientific model organisms, I think zebrafish are the simplest that show sort of abstract, anything resembling abstract learning.
I just realized that I think probably we all know something about our ancestry, that is the sort of 100 million years of being bacteria and things like that. And if you go backwards, there have been mammals for about 100 million years, I think.
and there's 100 million years of fish, and 100 million years of amphibians, and 100 million years of, those are all vertebrates, of mammals, of reptiles, and so forth. And I don't know what happens in the early period, except that we're descended from yeast somehow. And so it'd be nice to know what are the first few steps up to the worm, and where did it branch, and are we in that lineage, or did that lead off to the coelenterates and other things that we don't have any horizontal relation? Anybody know?
I'm pretty sure that we're not descended from C. elegans. I think that is a separate, the worms are a separate branch from the vertebrates.
But there must have been something like a paramecium.
Right. So
There's
really interesting work in looking at yeast that flock. There's in typical yeast, sort of wild type yeast, when it reproduces, the daughter cell sort of diffuses away. But there's you can get yeast to adhere to itself. So as it reproduces, it forms these globs. And it's actually been shown that under certain environmental conditions that are not that implausible, that those globs are more Darwinian fitness than individuals. And so there's a hypothesis that that's how yeast started to become multicellular.
So they must have some extra genes that are not activated normally. I wonder why it isn't taught in grade school. Is that because evolution is not allowed in? But I came from New York. There weren't many anti-evolutionists yet. Maybe there were. Well, I'm impressed. I think that sounds like a very exciting adventure.
Thanks.
Do any of you have a plan to pursue AI or psychology? Who has a career plan? I don't remember ever having one. There was just something exciting to do next week. Anyone have a criticism? Should David actually do this?
Well, biology is slow, and computers are fast, and they get faster. I think that if someone were to put as much work as David is putting into the biological aspect of this, to try to brute force a model of the worm faster than he could, and try to match it up with the behaviors, they would match up those 30 behaviors faster than we would.
I would love a competition. I would just like to point out that lasers are also fast.
Well, there's something wonderful about an animal that can reproduce in 4 days, because you could actually, as part of your 4 years plan, you could actually plan to breed some that have some particular new neurological behavior on the side.
Is there any kind of like social behavior that's like documented with the neurons, or is there like a small animal that you could, with social behaviors that you could study on this? Like interaction between.
Yeah, I know what you mean, and C. elegans doesn't. Well, actually, it's kind of funny because the hermaphrodite doesn't have any social behavior, but the male does, for obvious reasons. There's in fact 70 extra neurons in the male for the purposes of finding a hermaphrodite to impregnate. But I don't know, again, not being a zoologist, I locked onto this because it's well studied and it's the very simplest. But questions of the form, what's the simplest under constraint X, I'm less well equipped to answer. As far as I know, the best thing in that domain would be ants, but I'm sure there's something simpler than ants that exhibit social behavior. I just don't know what.
Well, isn't there some yeast that forms at some stage, it actually forms a sort of tower and stands up? I think it's yeast.
Well, yeast don't have neurons, so
Right.
these sorts of techniques won't apply there.
Slime mold, perhaps?
Yeah, maybe that's the thing.
Yeah. Yeah, slime molds are interesting because they're sort of all neuron in a way. They're just clumps of cells that happen to have electrical activity. There might be something interesting there. I don't know if anyone's tried to express optogenetic channels in slime molds.
They're very small.
They're very small, but you can do it with a virus, maybe.
Well, what about at a higher level? How would you find something like K-lines?
I think that you need some way of seeing a lot more things in a lot bigger of a brain. The one thing that comes to mind as sort of a new technique that might turn up K-lines is diffusion tensor imaging, which is a way of using MRI to find quite detailed structure. And I don't know the physics of it, but it involves following water molecules as they diffuse or tracking their flow.
Oh, so if something is more active, the diffusion is faster.
No, it's not functional. It's structural. It's that if there's a bundle of neurons, then the diffusion will be highly anisotropic, and you can measure the anisotropy, and then you can use some tensor math to turn that into what's called a tractogram.
But you're measuring the heat flow or something?
It's magnetic. It's MRI. It's magnetic resonance imaging. So it has some sort of tomographic component. And again, I don't know the physics of it. I just, in fact, heard of it a few weeks ago. It's a new technique, but it's been used to create some maps of the, particularly I think in monkeys, it's being used a lot. To create maps of at least the long-range connections between a whole lot of different areas. But even those are, it really can only resolve thick bundles of neurons, and K-lines probably aren't. But maybe they are. They might turn out.
I wonder how thin the skull of a parrot is. I just mention it because it might be the smartest animal per gram or something. Well, how smart are mice?
Mice are pretty smart.
Octopodes are supposed to be pretty smart too, and I don't know how much they weigh.
Which thing?
I think they're pretty heavy.
Octopodes are heavy. That makes sense. They're in water.
Right.
Dense.
I think you'd probably find ants to be the smartest for grad. But again, that's just my guess based on what little I know of animals.
Mm-hmm.
Parrots are always trying to optimize for weight, and ants aren't.
Ants are pretty small. Well, they're very variable, yeah. I think Ed Wilson had a 26-year-old ant.
It would be glorious to see a large group of ants acting as a parrot.
I just have a quick question for David. You said, well, there was an experiment that sort of showed that we could control mice by shining lights on them.
Yeah.
Do you think that there's any fear of the possibility that someone could create a virus that infects all humans and then it controls humans to do more than just turn right by shining lasers at them from space?
So the
Fear or hope?
what?
Fear or hope?
Yes. Yeah, technology is a double-edged sword. No, I mean, human skulls are pretty thick. And even my skulls are pretty thick. In order to make this happen, you have to have a hole in the skull, and you have to mount your LED in that hole. So there isn't, so far, any way to do this from a distance without having some sort of physical surgical operation.
Space is also far away.
Right, if you're inside a building, like, it probably won't work, and most important people are inside buildings when they're making important decisions.
Where you tit pat.
Yeah.
Yeah, I mean, it's, right now there isn't any fear of that, but it's certainly something to think about, you know, as technology gets better. You never know when we might cross that threshold, but I think for the next 10 years or so, we're probably pretty safe.
Well, there are microscopic parasitic worms, and so it would be hard to direct them to go anywhere particular, but
Right.
you could certainly evolve some that go into the brain and go somewhere without destroying anything important,
Well, there
and,
are.
and drop little packages here and there.
There are actually none that infect humans. Actually, there is one that infects humans that has a more subtle effect. But there's actually a whole class of parasites which do locate to the brain of larger animals and do, in fact, cause them to engage in behaviors that are suicidal for the host but beneficial for the parasite.
That's right. What's the one that causes some insect to climb up to the top of a tree and?
I don't remember the name of it.
But
I know that it exists.
It gets in the brain and makes it climb the tree and
Yep.
and then jump off or whatever, and that spreads this particular parasite.
Right.
There's an easy way to control.
So don't go outdoors.
There's also, what is it, Toxoplasma gondii, which was on crack.com, which causes mice to enjoy the smell of cat urine.
Right.
Yeah, it's probably one of the ones you're thinking of. So it'd be interesting to develop something like that for humans.
I heard people can actually get it from cats or something. Can it
Yeah,
taste
humans carry it, but and it does what was
That's
supposed to-
I think that's the one I was thinking of. It has a subtle effect in
It's
humans.
pretty benign on humans.
It's boring. It is in fact.
Like it makes girls more flirtatious or something.
Yeah, I think that's it. Yeah.
So attention.
Well, these are all instances of the future dangers of getting better scientific techniques for high school students to do experiments with.
There's an easy way to control people. Just throw money at them.
Just throw what?
Throw money at them.
And they will do whatever they want.
Oh, yeah.
Yeah, actually, that's one path that I forgot about, that some people do actually pursue. You can just reason directly from what makes people the most money. And that gets you something. It makes a lot of money. It's kind of like a person, I guess.
I think there are mutations of people that are not affected by money, though it would be interesting to try to develop a virus to counteract money.
Yeah, there's certainly
we can treat it.
We can treat alcoholism with disulfiram. So maybe there's some way that we can treat avarice.
That needs a downfall of our economy.
Yep.
What's this new alcoholism treatment?
Oh, it's not that new, but it's a drug that makes alcohol incredibly repugnant. And so it's sometimes given to people with severe alcoholism so that they are repulsed by alcohol and don't drink it anymore, as long as they continue to take the medication.
Mm-hmm. There's something like that in my family, because if I drink alcohol more than a small amount, then these little things like ants start crawling on my face, and they're very unpleasant. Any of you have that?
A lot of Asians have it, actually.
Really?
Or maybe it's not, the one that I have is like, you lack alcohol dehydrogenase, the thing that breaks down alcohol, and you have 2 copies of the gene. And I have like one good copy and one bad copy, depending on what you find is good or bad. And so I can drink like a glass, but any more than that, like I kind of get really itchy and like red.
Uh-huh. Yeah, so I don't have any.
But the people who have 2 copies where they lack the enzyme, then like if they have a sip, it's like bad things happen.
Oh. Well, evolution produces all sorts of strange things.
Neutral drift racing is still legal.
What?
Neutral drift racing is still legal.
What's that?
It's when an organism goes under neutral drift. It's 2 organisms competing to not change.
Yes, why hasn't the net been destroyed by a virus by now? Is there any?
Why hasn't what been destroyed?
The internet.
Oh, because it has white blood cells. the system administrators act as white blood cells, so they're
Kaspersky.
Yes. And it is
Bruce Steyer is the reason the internet is still here.
Well, it is largely immunized, but it's still infected by parasites. Like there are botnets, the size of which we can only estimate, that are sort of parasites running on the internet. Interestingly, analogous behavior.
Yeah, I just find it surprising that there hasn't been a really large disaster yet. Because
partially because it's not Xanadu, right? It's not Ted Nelson's super centralized internet. It's a distributed, redundant.
There have been a few attacks that took down large parts of the internet, but nothing quite took down everything.
say it again.
There were a few attacks that took down large chunks of the internet in the past.
Yes, I think Croatia's internet was taken down by a woman with a spade who was trying to mine some copper. Oh, this looks like a lot of copper. I better turn it
Well, I had a Microsoft virus for years, but it never did any harm. It just if I forget what it was called. If I got rid of it, it would come back again. And
people in Croatia still had internet access, though. They could connect via satellite.
This is true.
So the internet now has sort of enough redundant methods of making connections.
Not quite. People who have satellite links rarely share them.
Yeah, I guess the thing is that the initial statement is, why hasn't the internet gone down where the internet is whatever's is sort of defined as whatever's still connected to anything else, since
Right.
the internet is defined by its connections.
Right.
Well, why hasn't there been a smallpox epidemic that killed everyone? Because that has happened for quite a few species.
But all the species currently alive have not been killed by smallpox.
Right.
So,
That is correct.
The anthropic argument. Always correct and never quite satisfying.
I heard an hour-long program this morning about the, what's his name, on WBUR, about making high-speed trains in California. And it was all very interesting and incredibly expensive. And I wonder, and the current plan is to make one that'll take 30 years to construct, which seems that's rather odd, because you can't expect any particular government, including California, to be stable. But I wonder what's the point of people. Are people really going to travel at great expense and cost when they could have telepresence? And
Yeah, moving mass around is kind of a ridiculous way to transfer the information that's inside your brain, so it can be used in a meeting somewhere.
And at some point, we might just say, shouldn't we ban international travel? Just because of the danger of a plague. And I think the danger of a plague is going to suddenly increase because of high school students and doing science fair projects. Because the nice thing about evolution is it doesn't have, contrary to some beliefs, it doesn't have any intentional agents directing it. But once you can make gene strings in high school, then Darwinian evolution becomes a minority. And
I assume you've heard the recent news of Dutch biomedical engineers who produced a version of H1N1 with 60% mortality.
oh, and where does he keep it?
In his basement.
For science!
Did you make that up?
Well, I assume he did it for science. I made up the tone of voice.
Oh.
Was this a military-funded project?
No. This was at a hospital, at a hospital research institute. I mean, presumably, it was funded by someone who's interested in making a cure for H1N1, and they're trying to make it more obvious when you have one in your test population of chinchillas or whatever their model organism is.
Ferrets.
Ferrets. That's right. Close enough.
You knew what it sounded weird.
Okay. Well, next time bring some questions. Thank you.